Good morning, everybody. Today's class is about endocrine, minor endocrine glands. As you all know, the major minor endocrine glands are the thymus, the pineal gland, erythropoietin, atrial natriuretic peptide, and the local hormones. Thymus is very important because it is believed to be acting through a hormone called thymosin, which controls the development of lymphocytes and thereby responsible for cell mediated immunity. Located in the anterior part of the upper mediastinum, its weight reaches its peak during adolescence from 10 to 20 to 30 grams and then declines as with increasing age. Most of the cortex and medulla is replaced with connective tissue. The gonads exert a depressant effect on the thymus, that is, it involutes the thymus. It has a very clearly defined cortex and medulla. It is located in the mediastinum. The functions of thymus is that it provides a favorable environment for lymphopoiesis. It plays a very important role in processing of T lymphocytes. Thymus is the site of initial T lymphocyte differentiation. Immature T lymphocytes migrate from the red bone marrow to the cortex of thymus where they proliferate and mature. If the gland is removed during childhood, there is complete arrest of development of cellular immunity. The epithelial cells of thymus secrete thymosin, a hormone that controls the proliferation and maturation of primitive lymphocytes into immunological competent T lymphocytes. It also secretes thymopoietin, another hormone which is a polypeptide that induces the differentiation of pro-lymphocytes into immunological competent T cells. Medulla contains more T cells, and finally, the T cells that leave the thymus are carried to lymph node and spleen. The reticuloepithelial tissue of thymus secrete a hormone thymosin which stimulates lymphopoiesis both within the thymus and the peripheral lymphoid tissue. Thus, thymus promotes the development of immunologically competent T lymphocytes. Thymus also secretes thymopoietin or thymin, which inhibits acetylcholine release at the motor nerve endings in myasthenia gravis. Clinical importance is in children with genetic immunodeficiency disease and in patients with lymphocytopenia secondary to radiation and chemotherapy for cancer. Significant clinical improvement has been reported after injection of thymosin. The second is there is hyperplasia of thymus in autoimmune disease like myasthenia gravis, hemolytic anemia, and Hashimoto's thyroiditis. So, surgery in the form of thymectomy is normally done in patients with myasthenia gravis, and the prognosis is good, and there has been good results following the surgery. The pineal gland is very essential. It's another endocrine gland which is given a lot of importance earlier for the endocrine functions, has now been known to control the day-night cycle through secretion of melatonin. It's a small conical structure that is attached by the pineal stalk to the diencephalon. The gland is termed pineal gland because it resembles a pine cone. It is embryologically derived from the evagination of ependymal cells of the roof of the third ventricle and is located outside the blood-brain barrier. It lies in between the superior colliculi of the brain and has a very good blood supply. It is said to be a neuroendocrine transducer as it secretes a hormone called melatonin in response to sympathetic activity. The pineal glands lack the blood brain barrier, and therefore drugs and dyes penetrate rapidly into the pineal gland through poorly absorbed by the rest of the brain tissue. It acts as a biological clock which monitors sex glands. It is located on the roof of the third ventricle, as you see. It weighs around 150 milligram. Melatonin is synthesized from 5-hydroxytryptamine. These are all indole amines. The pineal gland not only secretes melatonin, it also secretes peptide hormones. It is synthesized from tryptophan, and the rate limiting enzyme is serotonin N-acetyl transferase. It is synthesized in the parenchymal cells, those are called as pinealocytes. There is a diurnal variation in the secretion of melatonin. Its synthesis and secretion are increased during dark periods, that is during night, and maintained at low levels during daylight hours. Melatonin secretion declines during the age. It's around 75 picograms per ml in adults and 250 picograms per ml in children. It plays a major role in skin color as it lightens the skin color. It plays an important role in regulation of circadian rhythm along with the hypothalamus. May have a role in the control of seasonal behavior like mating and hibernation in animals. It has an anti-gonadotropic action. Main action is on the anterior pituitary to inhibit the release of gonadotropic hormones. Pineal gland has a role in the regulation of onset of puberty. It is seen that precocious puberty occurs, that is, early puberty occurs in patients with pineal tumors that decrease melatonin secretion. In true pinealomas, where there is increased secretion of melatonin, delayed puberty and hypogonadism are seen. Puberty is also delayed in blind girls due to increased melatonin. The pineal gland may secrete a substance which delays the onset of puberty. It has a role in slowing the electroencephalogram rhythm, and it raises the convulsive threshold. The mechanism of action of melatonin is through MT1 and MT2 receptors. As it acts through the MT1 receptor, it increases the cyclic AMP, and through MT2 receptors, it increases the inositol phosphate 3 and diacylglycerol. Stimulation of the sympathetic supply to pineal gland releases epinephrine and norepinephrine, which in turn stimulates the melatonin synthesis by binding to beta adrenergic receptors. And this is the mechanism of action. It also has inhibitory actions on most of the endocrine glands like adrenal, thyroid, and pancreas. It inhibits corticosterone and aldosterone secretion. It inhibits thyroid secretion by inhibiting the TRH. It has a role in thermoregulation, and finally, it has an anti-tumor activity due to the antimitotic effect of melatonin. The other minor gland, as we have studied, is the erythropoietin, which is produced by the peritubular capillaries of the interstitial cells in the and liver. May also be produced abnormally by some tumors. Most potent stimulus for the release of erythropoietin is low PO2. The action of erythropoietin is mediated by mRNA synthesis. It acts on the erythropoietin sensitive stem cells in the bone marrow and increases red blood cell production. Factors that increase erythropoietin secretion include hypoxia, androgens like testosterone, growth hormone, prolactin, luteinizing hormone, glucocorticoids, and thyroid hormones. All this increase the erythropoietin secretion, whereas high PO2 and estrogen decrease the erythropoietin secretion. As you all know that during when we studied about erythropoiesis, it was the major role in regulation of erythropoiesis was by erythropoietin, which was synthesized from erythropoietinogen, also called as renal erythropoietic factor. It is regulated the rate of erythropoiesis mainly by the blood oxygen levels. Hypoxia stimulates due to decreased RBC and availability. Reduced O2 levels in the blood causes the release of erythropoietin. Erythropoietin stimulates the bone marrow and enhances erythropoiesis, wherein more RBCs are produced, which increases the oxygen carrying capacity. This is how the regulation. There is a balance between the oxygen levels. Normal oxygen, it is in this form. Whenever the oxygen decreases, that is during hypoxia. So hypoxia is a potent stimulus for release of erythropoietin. The next endocrine gland is the atrial natriuretic peptide, which is produced by the atrial muscle cells. Its action is mediated through cyclic GMP, and thereby it increases the GFR and renal blood flow. It also decreases the responsiveness of vascular smooth muscle to various vasoconstrictors. It inhibits renin and ADH secretion. With the above action, it decreases the blood volume and blood pressure. It is useful in the treatment of edema. This is the only hormone which received Nobel Prize in a single year. ANP is increased by increased sodium intake and by an increase in ECF volume. ACF is increased. ANP is also decreased when there is decrease in central venous pressure. The local hormones which we are familiar with from the time you join the MBBS are the substances which act at their site of synthesis and release are called local hormones. They are predominantly acetylcholine, histamine, serotonin, prostaglandins, adenosine derivatives like AMP, ADP, ATP, and plasma polypeptides like angiotensin, kinins, and finally the nitric oxide. Acetylcholine, you know, from the time we studied about neuromuscular junction or any, we have two types of receptors, the muscarinic receptors and the nicotinic receptors. The muscarinic receptors, these are all cholinergic receptors. The muscarinic receptors are present in heart, smooth muscle, and glands, and nicotinic receptors are present in the autonomic ganglion and the motor end plate of neuromuscular junction. Actions of muscarinic receptors are similar to alkaloid muscarine. The effects are blocked by the drug atropine. Brain contains both muscarinic and nicotinic receptors. Presynaptic nicotinic cholinergic receptors are present in the brain. They are located on glutamate secreting axon terminals. Stimulation of these receptors facilitates the release of glutamate into the synaptic cleft. Glutamate is responsible for 75% of excitatory transmission in the brain, and this is called as the excitatory drive in the brain. Are you in a state of excitatory or inhibitory? We are always in a state of inhibitory, or otherwise we have developed all sorts of neurological disorders. The next Local hormone is histamine, which is formed from the amino acid histidine by action of the histidine decarboxylase. The actions are it relaxes the arterioles and precapillary sphincter. It increases the tone of smooth muscle of intestine, bladder, and bronchioles. It is a potent stimulus of hydrochloric acid in the stomach and moderate stimulant for pepsin secretion. It plays a role in production of pain and itching. Its mechanism is through H1 and H2 receptors. Serotonin is a cardiac stimulant which is synthesized from tryptophan. It is a powerful vasoconstrictor. It increases systolic and diastolic blood pressure. It produces hyperapnea and increases tone of smooth muscle. Both histamine and serotonin were essential for triple response. Prostaglandins are membrane associated biologically active lipids synthesized by every mammalian cell and tissue. It is formed from arachidonic acid. It has wide variety of actions. On cardiovascular system, it produces vasodilation and it is useful in the treatment of hypertension. On reproductive system, it stimulates the gravid uterus and induces labor. It stimulates the release of GnRH and it is useful in MTP, that is medical termination of pregnancy. It causes bronchial relaxation and is useful in asthma. And it is a neurotransmitter. It also has action on the pupillary muscles, like thereby it produces meiosis, that is pupillary constriction, and it also increases the intraocular pressure. It stimulates the intestinal motility. There are various prostaglandin inhibitors like NSAIDs, which we call non-steroidal anti-inflammatory drugs like aspirin, indomethacin, steroidal anti-inflammatory drugs like hydrocortisone. The next is a lot of adenosine derivatives, and the most important one which we have all been discussing is acetylcholine, and this is a potent vasoconstrictor. You know that angiotensin is mainly synthesized from angiotensinogen. The actions are predominantly 40% pressor activity, that is vasoconstrictor, and 100% aldosterone activity. It stimulates the release of ADH and ACTH. And the last is the nitric oxide, which is a vasodilator. In clinical importance, we use it for angina, unstable angina in the form of NTG, that is nitroglycerol. Thank you. We finished endocrines and you'll have your assignment. Please submit the assignment.